about coated vesicles. Vesicles are used to transport proteins both out of the cell and into the cell. The secretion of proteins is called exocytosis. Internalization of proteins is called endocytosis. Proteins are transported in coated vesicles. Constitutive transport from ER through the Golgi takes place by COP, coated vesicles. Clathrin coated vesicles are used for both regulated exocytosis and endocytosis. Let's know the key points. Protein transport through the endoplasmic reticulum Golgi system occurs in coated vesicles. Secreted proteins move in the forward anterograde direction toward the plasma membrane. This is also movement within the system in the reverse retrograde direction. Proteins that are imported through the plasma membrane also are incorporated in coated vesicles. Coated vesicles bought from a donor membrane surface and fused with a target membrane surface. Endocytic and secretory vesicles have clathrin as the most prominent protein in their coats and are therefore known as clathrin coated vesicles. The subunit of coat consists of a triskeleton, a three prolonged protein complex. The inner cell of the coat is made by proteins called adapters, which bind both to clathrin and to integral membrane proteins of the vesicle. The inner cell of the protein of the coat is made by proteins called adapters, which bind both to clathrin and to integral membrane proteins of the vesicles. COP, COP, eye-coated vesicles are responsible for retrograde transport from the Golgi to the ER. COP2 vesicles undertake forward movement from the endoplasmic reticulum to Golgi apparatus. In this pathway, for regulated secretion of proteins are sorted into clathrin-coated vesicles at the Golgi transphase. Some vesicles may fuse into secretory granules. Vesicles also move to endosomes, which control trafficking of the trafficking to the cell surface. Secretory vesicles are stimulated to unload their cargoes at the plasma membrane by extracellular signals. Similar vesicles are used for endocytosis, the pathway by which proteins are internalized from the cell surface. Now let's know about electrical properties of membranes. All animal cells are surrounded by a plasma membrane. The fluid on both sides of the membrane contains high concentrations of mobile ions, of which sodium Na plus and potassium K plus, chlorine Cl minus and calcium Ca2 plus are most important ions. The membrane potential the membrane potential arises from the interaction of ion channels and ion pumps embedded in the membrane, which maintain different ion concentrations on the intracellular and extracellular sides of the membrane. The membrane potential has two basic functions. First, it provides power to operate a variety of molecular devices embedded in the membrane. Second is in electrical excitable cells such as neurons. It is used for transmitting signals between different parts of the cell. Opening or closing of the ion channels in the membrane produces a local change in the membrane potential, which causes electric current to flow rapidly to other points in the membrane. Let's know about exocytosis and endocytosis. Let's know first of all exocytosis. During exocytosis, vesicles often formed by the Golgi apparatus and carrying a specific molecule fused with the plasma membrane as secretion occurs. This is the way that insulin lives in insulin secreting cells. For instance, notice that the membrane of the vesicle becomes a part of the plasma membrane. During cell growth, exocytosis are probably used as a means to enlarge the plasma membrane whether or not secretion is also taking place. Endocytosis. Let's know about endocytosis. During endocytosis, cells take in substances by vesicle formation. A portion of the plasma membrane invaginates to envelop the substance, and then membrane pinches off to form an intracellular vesicle. When the material taken in by endocytosis is large, such as food particles or another cell, the process is called phagocytosis. Phagocytosis is common in unicellular organisms like amoebas and in amoeboid cells like macrophages, which are large cells that engulf bacteria and own out red blood cells. In mammals, when the endocytosis vesicles fuse with a lysosome, digestion occurs. Pinocytosis occurs when vesicles form around a liquid or very small particles. Blood cells that line the kidney tubules or intestinal wall and plant root cells all use this method of ingesting substances. Whereas phagocytosis can be seen with the light microscope, the light microscope must be used to observe pinocytosis, pinocytic vesicles, which are no larger than 1 to 2 micron. Phagocytosis occurs when the substance to be transported into the cell is large. Certain specialized cells in the body can engulf worn out red blood cells by phagocytosis. Digestion occurs when the resulting vacuole fuses with a lysosome. Pinocytosis occurs when the macromolecule such as polypeptide is to be transported into the cell. The result is a small vacuole or vesicle. Receptor mediated endocytosis is a form of pinocytosis. The substance to be taken in a ligand first binds to a specific receptor protein which migrates to a pit or is already in a pit. The vesicle that forms contains the ligand and its receptor. Sometimes the receptor is recycled, and receptor mediated endocytosis is a form of pinocytosis that is quite specific because it involves the use of receptor proteins shaped in such a way that a specific molecule such as vitamins, peptides, hormones, and lipoproteins can bind to it. The binding of a solute to the receptors causes the receptors to gather at one location. This location is called a coated pit because there is a layer of fibrous protein on the cytoplasmic side. Once the vesicle is formed, the fibrous coat is released and the vesicle appears uncoated. The fate of the vesicle and its contents depends on the kind of solute it contains. Sometimes the solute simply enters the cytoplasm. A spent hormone, on the other hand, may be digested when the vesicle fuses with a lysosome. The membrane of the vesicle and therefore the receptors are returned to the plasma membrane or the vesicle can go to other membranous locations. Aside from simply allowing substances to enter cells selectively from an extracellular fluid coated pit are also involved in the transfer and exchange of the substance between cells. Such exchanges take place when the substances move from maternal blood into fetal blood at the placenta, for example, the importance of the receptor mediated endocytosis is demonstrated by a genetic disorder called familial hypercholesterolemia. Cholesterol is transported in blood by a complex of lipids and proteins called low density protein LDL, low density lipoprotein LDL. These individuals have inherited a gene that causes them to have a reduced number and or if defective receptors for LDL in their plasma membranes. Instead of cholesterol entering cells, it accumulates in the walls of arterial blood cells, leading to high blood pressure, occluded arteries, and heart attacks. Thank you.